Good afternoon, everyone, and I'm Elita Montanari, the CTO and acting CEO of EVis Bioscience, which is an ETH spin-off that was incorporated in June 2024. So we are a young company. EVis Bioscience is based near Zurich in the biotech campus in Schlieren. So RNA-based drugs might have a tremendous therapeutic impact. RNAs might be used for several medical applications that do not have a therapy yet. For example, they could be exploited for protein replacement as vaccines, for cancer immunotherapy, for cellular reprogramming, and for gene editing. RNAs and DNAs need a carrier to be delivered and protected from degradation into our bodies. So far, only two delivery strategies have been approved by the FDA. Synthetic lipid nanoparticles that are delivering messenger RNA that we can find in the anti-COVID-19 vaccines from Moderna and Pfizer, and viral particles for the delivery of DNA in the nucleus of cells. However, lipid nanoparticles cannot target specific cells or tissues and can be toxic after repeated injections. On the other hand, viral vectors are immunogenic and have low packaging capacity. So this means that big genes cannot be packed into viral vectors. To overcome these limits, extracellular vesicles, shortly EVs, have been investigated for 20 years also in gene therapy. EVs are biological carriers that naturally deliver RNAs and proteins into our bodies. And depending on the selected EV, a different cell or tissue can be targeted. And this is very important for gene therapy because we want to deliver the RNA where is needed. So the main advantages of EVs are the biocompatibility because they are coming from our body and the targeting capability. However, they also face relevant limits, such as the scalability of the isolation and purification processes of EVs and the loading of EVs with exogenous and therapeutic RNAs without destroying the vesicle structure. So EVis Bioscience is the first company to employ a unique approach to quickly and efficiently load RNAs into EVs, forming these hybrid extracellular vesicles, shortly hybrids, offering novel opportunities for an extrahepatic delivery. So it means for targeting tissues that are out of our liver. Here we can see the morphology of hybrids derived from three different sources from the human body. So red blood cells EVs, mesenchymal stem cell EVs, and plasma. And we can see how the LNV is which is carrying the RNA, is hemifused to the EV surface. These carriers can provide specific targeting depending on the selected EV. More than 90% of messenger RNA can be loaded into our hybrids, and the hybrids loaded with messenger RNAs can transfect cells and express the protein of interest. Further, hybrids loaded with short interfering RNAs can silence genes in cells. So the EVis Bioscience business strategy is very versatile and mainly relies on three pillars and maximize both scientific and social benefit. The development and commercialization of the EVis NanoVector to university and research institutions to support their research programs in the EV field that will be ready by mid-2026. The development of the hybrid carrier that can be licensed to pharma companies to expand their therapeutic pipeline, and the development of a proprietary therapeutic pipeline for treating rare genetic disorders. So between 2022 and 2025, big pharma companies, including Novartis, Pfizer, and AstraZeneca, acquired smaller companies developing gene-based therapeutics, almost all in a preclinical development, offering upfront payments of hundreds of millions. Two patent applications protect our technology. The first patent was filed in September 2023 and protects the chemical composition of LNVs and the hybrids. The second patent application was filed in February 2025 and extends the protection of the chemical composition of the hybrids and their medical applications. So EVis already won the Venture Kick phase 1 and phase 2, the innovation check, and just a few days ago we also won the InnoSwiss with implementation partner, which is a competitive and very important Swiss grant. And this grant will financially support the preclinical research until 2027 and will allow the EVis team to expand. From mid-2026, the European pre-accelerator and the chemical kits with the sub-licenses will drive EVis to the 2027 when we will start to prepare the clinical batch, and then the InnoSwiss starting grant and eventually Venture Kick will carry EVis until 2028 when we are considering to make an exit after an IND or CTA. So the budget obtained will allow in 2020, sorry. So the amount of money that needed by the EVis Bioscience to achieve the IND or the CDA is almost 12 millions, and planned inflows exceed these by almost 1 million. Our goal with Capital Cell is to raise 700 euros, and which will be used primarily to conduct preclinical investigations and to develop and commercialize the EVit platform. And in 3 weeks, we already secured more than 70%, and the campaign will continue for more than a month. So significantly, the company's potential value could increase approximately 25 times by 2028. So the management team combines scientific, business, and financial expertise. EVis is further supported by a panel of experts in the pharma and clinical fields, and the CEO with a deep pharma experience will be hired by 2026. So the EVis technology already received financial support from several Swiss and European grants, and the company has ongoing scientific collaborations with international universities and experts worldwide. So if you would like to have more information about EVis, just follow us through the website, the LinkedIn page, and you can text us to our email. And thank you very much for your kind attention.